Session will be chaired by Professor Dr. Pratyut Prakash, Department of Microbiology, Institute of Medical Sciences, Banaras Hindu University, Varanasi. We welcome you, sir. And co-chaired by Dr. Amresh Kumar Singh, Professor and in charge of HICC AMR Department of Microbiology, Baba Raghav Das Medical College, Gorakhpur. We welcome you too, sir. And assisted by Dr. Kiran Kumar as rapporteur. Thank you. We extend a warm welcome to our distinguished panel and invite all participants to engage attentively in this session. Now hand over to you, sir.
Attention please. Kindly be seated earliest so that we can start the session timely. So, good morning to all of you, and with this responsibility of co-chair of this session, I would like to introduce Dr. Deepa Srivastava. She is assistant professor in the Department of Botany at Gorakhpur Vishwavidyalaya, Gorakhpur, and she previously served as a UGC post-doctor fellowship in the same department, and she has many publications and I think 42 publications in reputed and national as well as international journal, and she is expert in her field, and she will deliver the topic microbiome derivative antimicrobials, a local strategy to combat AMR across community and hospital setting. So I would like to welcome you, ma'am. Please, dais is yours.
Thank you, sir. Hello. Hello. Hello, good morning, good afternoon to everyone. I am Dr. Deepa Srivastava from Department of Botany, DDU Gorakhpur University. Today I will deliver my talk on microbiome-derived antimicrobial, a local strategy to combat AMR across community and hospital settings. Screen. इसे प्लीज आप बता दीजिए कैसे ये फॉरवर्ड होगा. Introduction to AMR, a global health emergency. Antimicrobial resistance is the ability of microorganism, that is bacteria, fungi, viruses, parasite, to survive exposure to previously effective drugs. According to WHO, AMR is one of the top 10 global public health threats. Currently, more than 1.27 million deaths annually are directly attributed to resistant infection. Next. What is microbiome? Microbiome is fungal component of microbiome present in soil, plant, human, hospital surfaces. It is also the fungal component of microbiome resistant in gut, skin, oral cavity, lungs. Thank you. Environmental leaks. Major genera are Candida, Aspergillus, Saccharomyces, Melsesia, and Clostridium. Now, Why focus on microbiome? Because it produced diverse secondary metabolite, historically yielding life-saving antibiotics, penicillin and spironolactone, vast and explored indigenous floral diversity, local ecological adaptation mean unique bioactive compound. Fungi constitute 38% of natural product, antimicrobial, revealing bacteria in bioactive diversity. Microbiome interaction shape host immunity and pathogen resistance. Emerging One Health view link community and hospital AMR to microbial ecology. Microbiome derived antimicrobials are bioactive compound isolated from human commensal fungi, environmental fungi, hospital associated fungal community. These compounds may inhibit bacterial growth, disrupt biofilm, interfere with quorum sensing, induce oxidative stress, and target novel cellular pathway. Unlike classical antibiotics, many fungal metabolites have unique chemical scaffold that have not previously encountered, reducing pre-existing resistance. Now, source of microbiome. Basically, they are agriculture soil, forest ecosystem, medicinal plant endophytes, the hospital environment isolate, wastewater ecosystem. These environment exist selective pressure that derive metabolite diversity, making them promising antimicrobial source. Why fungi are powerful? You all know that fungi produce secondary metabolite, polyketides, non-rhizosomal peptides, terpenoids, and alkaloid. The reason is that the fungi compete in microbial ecosystem, produce antimicrobial compound for survival. Antimicrobial wealth of fungi, Basidomycetes produce compound active against gram-positive and gram-negative bacteria. We have basically worked on Basidomycetes. Mushroom extract show strong inhibition of Staphylococcus aureus and E. coli and pathogenic fungi. Metabolites such as benzoxanoid, terpenoid, and lipopeptinoid target cell wall DNA and metabolism. Bioactive compound from fungi. These are major class of polyketides, alkaloid, terpenoids, peptides, phenolic compounds. Their activities are antimicrobial, antifungal, antiviral, antibiofilm, quorum sensing, and inhibition. Today I am concentrating on antibiofilm and anti-quorum sensing. These compounds target diverse pathway, reducing the chance of rapid resistance development. Target AMR pathogen, and Actinobacterium bromai and Pseudomonas aureus. Hospital-associated methicillin-resistant Staphylococcus aureus, MRSA, and CRE, and community-associated extended-spectrum lactamase, producing E. coli, drug-resistant Salmonella, and Candida aureus. The aim is dual impact, hospital-acquired infection, and community-level resistance. Why AMR in class 10? Because of over-prescription of antibiotics, self-medication, especially in developing country, incomplete treatment course, excessive use in livestock or agriculture, poor infection control in hospital, and environmental dissemination through wastewater. This is a timeline showing some of the key antibiotic discovery. First of all, penicillin was discovered. It was a fungal component, and even within 1 or 2 years, penicillin-resistant Staphylococcus was discovered. Then tetracycline in 1950 and 1959, tetracycline-resistant Shigella was discovered. Methicillin and methicillin-resistant Streptococcus in 1962. In 1967, gentamicin, and in 1980, gentamicin resistant Enterococcus. In 1972, vancomycin, and in 1990, vancomycin resistant Enterococcus in 1988, and in 1996, levofloxacin, and its resistant was developed by Streptococcus resistance in 2011, and this is the timeline in which we are seeing that most of the antibiotic which was produced, its resistance was developed within 2 or 3 years. Now, mechanism of bacterial resistance. It is number one is enzyme degradation. Example, beta-lactamase hydrazide, the beta-lactam ring in activating penicillin and cephalosporin, carbamatenes, Calvacia, pneumonia, and carbamatease, and NDM1 degrade last resort antibiotic. Efflux pump, membrane protein activity pump antibiotic out of bacterial cell. Example, ACR, ABN 12-1C, system of gram-negative bacteria. This lead to multi-drug resistance. Then target modification, bacteria alter antibiotic binding sites by altered penicillin binding protein and ribosomal mutation preventing macrolides binding. Reduce permeability, loss of modification of pore channel, prevent antibiotic entry and biofilm formation. Biofilm are structured and microbial communities embedded in extracellular polymeric substances. Biofilm reduce antibiotic penetration, slow bacterial metabolism, increase gene transfer, enhance resistance 10 to 100 to 1000 fold compared to planktinoids. The critical point is that most conventional antibiotic target limited pathways, cell wall, protein synthesis, DNA replication, resistance involved rapidly. Now, mechanism of action, cell wall. This is mechanism of action of fungi. The number one is cell wall disruption. Certain fungi metabolite inhibit peptidoglycan synthesis, compromise bacterial structural integrity. Membrane permeabilization. Some compounds insert into bacterial membrane, forming pores, ion leakage, and cell death. Reactive oxygen species induction. Fungal metabolite may increase intracellular ROS level, leading to oxidative damage of DNA and protein. Biofilm inhibition, some fungal component interfere with quorum sensing molecule like AHL, preventing biofilm. This is critical in catheter infection, implant infection, and chronic wound infection. Now, mechanism of action, we will discuss this in detail. Mechanism 1, cell wall synthesis inhibition. The concept is that fungal metabolite interfere with peptidoglycan synthesis, weaken bacterial structure integrity. The flow chart shows that first fungal metabolite penetrate bacterial cell, then bind to transpeptidase, inhibit peptidoglycan cross-linking, weak cell wall, and osmotic lysis, then cell death. The molecular explanation is that bacterial cell wall, that is NAG plus NAM chain, cross-linking of PBs if cross-linking is stopped, structure collapsed and especially effective in gram-positive bacteria. Now, mechanism 2, membrane disruption or pore formation. The concept is that some fungal peptide behave like antimicrobial peptides, amphipathic fungal peptides, and insert into bacterial membrane and oligomerize into barrel stave, pore, and carpet type disruption. This flowchart is that fungal lipopeptide, then electrostatic attraction of bacterial membrane, insertion of lipid bilayer, pore formation, then leakage of ions and membrane depolation, then death. Then molecular explanation, bacterial membrane are negatively charged, fungal peptides are positively charged. Electrostatic interaction allow intercession in ion imbalance, ATP depletion, and apoptosis like death. This mechanism is powerful because it acts as physically and resistant and difficult to develop. Next, mechanism 3, reactive oxygen species induction. Some fungal metabolite induce oxidative stress inside bacteria. This is that fungal metabolites interfere with electron transport chain, superoxide is formed, and hydrogen peroxide, then hydroxyl radial, and DNA damage and protein oxygen leads to cell death. Molecular explanation, ROS damage, DNA, membrane lipid and enzyme. Overwhelm, bacterial antioxidant defense, catalase and SOD. This is particularly useful in against MDR strain. Mechanism 4, biofilm inhibition, quorum sensing and interference. Fungal quorum sensing molecules such as farnesol can modulate bacterial behavior. They interfere with bacterial autoinducer signaling, inhibit biofilm formation, and increase susceptibility for antibiotic. Farnesol both disrupt membrane integrity of bacteria at higher concentration and downloads biofilm-related gene expression, reducing EPS production and biofilm robustness, a key advantage for tagging device associated with chronic infection. The flowchart shows that bacterial population produce autoinducers, AHLs, then quorum sensing activation, then biofilm gene expression, and EPS matrix formation. Now when we introduce fungal metabolite, fungal compound block AHL signaling, no quorum sensing activation, no EPS production, and weak biofilm. Increase antibiotic sensitivity and scientific importance. Biofilm are responsible for catheter infection, implant infection, and chronic wound. Biofilm inhibition is a major breakthrough area in AMR research. Mechanism 5, DNA replication inhibition or topoisomerase inhibition. Some fungal polyketides or alkaloid metabolite inhibit topoisomerase, DNA gyrase, or otherwise interfere with DNA replication machinery. This results in replication arrest, cell cycle blockage, and eventually cell death. New fungal compounds that target type 2 topoisomerase may offer against strain resistant to classical quinolones if they bind to alternate site or use different inhibition modulators. The flowchart is fungal alkaloid binds to DNA gyrase, prevent DNA supercoiling, replication arrest, cell cycle block, and cell death. Now mechanism 6, this is multi-target and antiviral section, local fungal isolate, secondary metabolite production, and active fungal compound. Multi-target bacterial attack, cell wall inhibition, then peptidoglycan cross-linking, structural inability, then membrane disruption, pore formation, ion leakage or ATP collapse, DNA protein damage, and then quorum sensing fails, topoisomerase inhibition and replication arrest, then all cell signaling EPS gene disrupt, and biofilm inhibition. These all, because of this process, metabolic collapse and oxidative stress begin, and high ROS and apoptosis begin. Now, why local matters? Regional AMR ecological rationale. Antimicrobial resistance show a strong geographic variation driven by local prescribing infection control, agriculture, antibiotic use, and environmental contamination. Recent global analysis and WHO reporting show rising regional burden and hotspots, especially in South Asia and part of Africa. Local fungal community, the microbiome, co-evolve with local bacteriomes, and environmental selective pressure. Those fungi are more likely to produce metabolite effective against locally dominant resistant phenotype. Therefore, local bioprospecting and targeted screening increase the chance of finding clinically relevant antimicrobials and improve translational fit for nearby hospitals community. Anti-biofilm potential. This is biofilm are major driver of hospital AMR. Fungal metabolite inhibit biofilm disruption mature biofilm, and enhance antibiotic susceptibility. Application in catheter and implant ICU surface. Biofilm derived hospital resistance. Fungal metabolite may act as antibiofilm agent or antibiotic adjuvant. Translational pathway in hospital application, antibiofilm catheter implant coatings incorporate identified fungal metabolite or non-toxic analog into polymer coating and test biofilm formation in vitro and in vivo. Surface disinfectant or ICU sanitation agent, develop formulation, stability, contact time, material compatibility, and compare with the standards disinfection, sodium hypochlorite quantity. Wastewater intervention, apply fungal metabolite cocktails to hospital effluent to reduce viable MDR, bacterial load, and ARG. Transitional pathway in community intervention, topical formulation of wound care that target biofilm common community pathogen. Water sanitization, point of use of solution of rural clinics to lower infection risk without systemic antibiotic resistant. Veterinary and agriculture uses to reduce prophylactic antibiotic use and environmental selection for ARGs. Integrating with One Health AMR management, human health, animal health, environment health, and plant pathogen and human pathogen interface. AMR is interconnected across ecosystem. Microbiome research aligned with One Health principle. Align with local AMR stewardship, reduce antibiotic usage where microbiome produced are deployed, and monitor resistance shift post-deployment. Advantage of microbiome strategy, novel chemical diversity, sustainability, local scalability, and lower resistant development risk, and eco-friendly. Challenges: standard standardization of extract, toxicity assessment, regulatory approval, scale-up formulation, and stability clinical validation. Future direction: metagenomics and metabolomics interaction, genome mining and cryptic biosynthesis gene cluster, community-based AMR surveillance, and nanotechnology-based delivery system. This was the concept, and what we have done in our lab, the workflow was the collection of fruiting bodies of macrofungi and their extraction, then isolation of compound having pharmaceutical properties, and identification of bioactive compound from HPTLC, qualitative analysis, and evaluation of pharmaceutical properties, antimicrobial activity, MIC value, antioxidant, DPPH scavenging assay, and IC20 assessment using bioindicator strain, chromobacterium virusium. This was the process system. First, we have taken Wolveria, Wolveria, powdered samples, oxalate extraction, and crude extract were prepared, and application was extracted done for different methods. Here only I will take biofilm formation method. The sample, this was the identification of Wolveria vellosa. It is called peristro fungi, and this is the antimicrobial activities. With different, the control was cephalosporin and Escherichia coli, staphylococcus were taken as microorganism. Test for antiquorum activity and extract. Screening of antiquorum sensitivity was done in NBRI, and screening of antiquorum sensing activity of the extract, Chromobacterium violatium ATCC 12472. They have done disk diffusion assay, violation production assay, phycocyanin production inhibition assay, and biofilm formation inhibition and eradication assay. This was the screening of antiquorum sensing. The screening of antiquorum sensing activity extract of Chromobacterium violatium. The quorum sensing inhibition was detected by a colorless, opaque, but viable halo around the disc. This was shown that the Wolveria has some activity which show quorum sensing, and it is a good candidate for antibacterial activity. Violation production assay, Blosser and Gray's technique was used to extract violation pigment. The dark purple color of its colony is due to production of insoluble pigment violation. And this was phycocyanin induction inhibition assay. This was the treatment, and this was the result. Phycocyanin production inhibition assay with treatment of ethyl acetate fraction of mushroom. It is ethyl acetate extract, and after treatment, the PSME against P. odiosa produced phycocyanin pigment was extracted from the supernatant with adding chloroform ethanol 2:1 followed by 0.2 HCl. After that, color of phycocyanin is changed from bluish green to pink color, and take absorbance at 520 nm. This is anti-biofilm activity of Pseudomonas aureus PA01, and we have seen that untreated and treatment, increasing the treatment, the anti-biofilm activity has increased. Biofilm inhibition and eradication assay, treatment of ethyl acetate fraction of Wolveria violacea against Pseudomonas aureus. And in conclusion, we can say local microbiome is strategy to combat AMR. AMR is region-specific and driven by local antibiotic use, hospital ecology, and environment reservoir. Local fungal microbiome, biodiversity is an unexplored source of novel antimicrobial secondary metabolite. Fungal metabolites act through antimicrobe and multi-target mechanism like cell wall inhibition, membrane disruption, ROS induction, DNA interface, and biofilm and quorum sensing inhibition. Integral local AMR surveillance plus fungal bioprospecting enable regional specific antimicrobial discovery. Application include ICU biofilm control, catheter coating, disinfectant, wastewater treatment, and community level tropical therapy. Supports One Health framework and reduce dependence on conventional bioactivity. A localized microbiome derived approach provides a sustainable precision-based solution to combat AMR in both hospital and community setting. These are some references. And we acknowledge HOD, faculty members of all botany department, faculty of CSIR and BRI, and our research team. This is our research team. Basically, this is the work is of Manisha Mishra. Thank you.
Thank you, ma'am, for your comprehensive and thought-provoking lecture. And sorry.
Now the house is open for discussion. So any query from the audience? One query from my side, and that is, you are advocating local microbiome and treatment options from those microbiome for the local isolate.
Hmm.
For this, then the policy makers has to validate individual claim from each region of the country. And as our own country has got a very diverse environment, so do you think it is feasible for the policy makers to validate each and individual claim?
No, it is difficult but feasible. If they, it will take more effort, but it is feasible and possible at least. And maybe there may be some common agent also. But we can prefer it will be quite.
But it is interesting to note that these fungal metabolite has got a very good anti-quorum sensing effect.
Yes.
So indirectly, it is anti-biofilm effect. So these can be, I think, in my view, it will be applicable for all the isolates,
Maybe.
not restricted to a region.
Yes.
So if a fungal isolate has got metabolite to inhibit quorum sensing agents, then it will be useful for not for the region, but I think for the entire bacterial kingdom.
Yes. Thank you, sir. Thank you so much, sir. Thank you.
Yes, one question from the audience. Yeah.
Good afternoon, ma'am.
Good afternoon.
Ma'am, what is quorum sensing mechanism?
Quorum sensing mechanism is actually the bacteria form a biofilm, okay, and due to which actually it is more effective in causing antimicrobial resistance. So if we disrupt that film, and quorum sensing actually bacteria communicate through it. बायोफिल्म एंड जैसे हम लोग बात करते हैं ना बैक्टीरिया बात करते हैं किसी जगह पे तो दिस इज एक्चुअली देयर कम्युनिकेशन तो इफ वी control the communication link or formation of biofilm, then we can reduce the antimicrobial activities, bacterial activity. We can reduce it. This work is for this only that we can reduce its quorum sensing, and fungi has the capacity to reduce quorum sensing and reduce biofilm formation.
Thank you, ma'am.
Okay, thank you.
So by inhibiting the quorum sensing means bacteria how talk to each other. When there is a adverse environment or the condition or a stress condition, bacteria will immediately talk to their neighboring bacteria. So this is the talking method, or you can say communication between two bacteria. So that is quorum sensing. And when we block the quorum sensing, so bacteria will not get aware that something is going to harm them. So by this mean, by her novel this work, we can block the quorum sensing, and that's why to inhibit the bacteria very easily, right? So any one question, I think we can take more questions. If no question, so thank you, ma'am.
Thank you, sir. Thank you. Sabhi ko shukriya. Thank you, sir.
Thank you, ma'am, for your wonderful lecture. And as a token of gesture, we request our chair and co-chair to kindly felicitate Dr. Deepa Srivastava with a memento.
Now the next speaker of the session is Professor Sarath Kumar Misra, who is Professor of Biotechnology and former Head of Department at DDU Gorakhpur University, Uttar Pradesh. With over 16 years of his teaching experience and extensive research experience, he has significantly contributed to biotechnology, molecular biology, and enzyme research. His research focuses on industrial enzymes and actinomycetes, derived antimicrobial, endophytes from medicinal plant, and phytochemicals for anti-cancer and antiviral properties. He has guided several PhD and master's theses. He has published widely in reputed journals, contributed book chapters with international publishers. He has been honored by academic and professional bodies. Professor Misra remained committed to advancing biotechnology for human welfare, environmental sustainability, and healthcare innovation. Today's lecture topic is novel arsenic-resistant bacterial strains from arsenic-contaminated regions of eastern Uttar Pradesh for arsenic bioremediation. Sir, now the stage is yours. Please.
Thank you so much, sir, for nice introduction. So, with permission from the chair, Professor Vidyut Prakash ji and Professor Amresh ji. So, after listening such a nice lecture by Dr. Deepa ji, I hope that all students, सब लोग सामने देखिए, मोबाइल खोले हो तो बंद कर दीजिए. मोबाइल वही लेकर खोलेगा जिसको फोटो लेना होगा मेरा. बाकी सब लोग मोबाइल बंद कर दें, ठीक है? क्योंकि अभी सर बता रहे थे सुनील जी की कैमरा लगा हुआ है एंड यू आर अंडर विजिलेंस, तो दैट कैन अफेक्ट योर मार्क्स. मुझे कुछ नहीं लेना देना, मैं तो आपको सलाह दे रहा हूं कि आपका मार्क्स अफेक्टेड हो सकता है, इसलिए मोबाइल रख दीजिए, आपस में बात मत करिए. अभी अमरीश जी बता रहे थे कोरम सेंसिंग, दिस वाज फॉर बैक्टीरिया, नॉट फॉर यू. So after entering in this hall, you should try to focus. I know you cannot focus. Try करोगे लेकिन फोकस नहीं कर पाओगे. 2 मिनट सुनोगे फिर दिमाग कहीं और चला जाएगा. होता है सर, इसीलिए समझ में नहीं आता है. समझ में नहीं आता है तो फिर क्या होती है, बोरियत होती है और जबरदस्ती बैठना पड़ता है. इसलिए ट्राई टू फोकस. दिमाग जाता है फिर वापस लाइए, फिर जाएगा फिर वापस लाइए, ठीक है? इस तरह से आप प्रैक्टिस करिए. तो ट्राई टू बी फोकस. जिसको बात करना होगा लेक्चर के बाद हम लोग बात कर लेंगे. शुरू करें? आर यू रेडी सब लोग? अच्छा, तो मैं कुछ बहुत बड़ी चीज बताने नहीं जा रहा हूँ, मैं ऐसी चीज बताने जा रहा हूँ जो आपके काम की है. दिस इज रिलेटेड टू आवर हेल्थ, ठीक है? आर्सेनिक का नाम सुना होगा आपने. तो बहुत सारे स्टूडेंट्स दे आर कमिंग फ्रॉम रूरल एरियाज or even from urban areas. Arsenic is in water, arsenic is in soil. जो पानी हम पी रहे हैं, क्या पता कि उसमें आर्सेनिक हो. बहुत अच्छी लाइफ जी रहे हैं, बहुत सिंपल लाइफ जी रहे हैं, एक्सरसाइज करते हैं, बहुत ही सिंपल खाना खाते हैं. उसके बाद यदि हम पानी में हमारे आर्सेनिक है तो हम बीमार पड़ जाते हैं. और आर्सेनिक से कैंसर भी हो सकता है. इसलिए इसके बारे में अवेयर होना, जागरूक होना, दिस इज माय मोटो ऑफ दिस टॉक. आर्सेनिक केमिकल एलिमेंट होता है एंड दिस इज नोन एज अ ह्यूमन कार्सिनोजन. कैंसर एक ऐसी बीमारी है जिसका नाम सुनते ही आधी मौत तो तभी हो जाती है. यदि किसी को बता दो कि कैंसर हो गया तो हाफ डेथ. इसलिए हम लोग बहुत डरते हैं इससे. तो हमें बचाव सबसे बहुत, प्रिकॉशन इज बेटर देन क्योर. तो किस तरह से हम बच सकते हैं? तो आर्सेनिक की बात कर रहे हैं. आर्सेनिक के ये बहुत सारे ऑक्सीडेशन स्टेट्स हैं, +3, 0, -3, +5. ये जो 2 मेन है आर्सेनिक के, आर्सेनाइट एंड आर्सेनेट, दे आर द 2 टॉक्सिक कंपाउंड्स ऑफ आर्सेनिक फाउंड इन द सरफेस वाटर एंड ग्राउंड वाटर. जो पानी हम पीते हैं उसमें ये 2 फॉर्म्स ऑफ आर्सेनाइट और आर्सेनेट यही नुकसान करते हैं. This is reported from several places like India, Bangladesh, Myanmar, USA, Canada, and China, and all those places. This is arsenic. बात यह है कि what is the permissible limit? कोई भी चीज जब एक लिमिट्स क्रॉस करती है तो वो हमारे लिए टॉक्सिक हो जाती है. What is the toxic level of arsenic? And for that, 0.01 milligram per liter means 10 ppb is the permissible limit. उससे ज्यादा होने पर, और जो permissible limit है that is given by different agencies, and this is different for soil and different for water. सोर्स कहां से आता है आर्सेनिक? दो सोर्सेज हैं. वन इज अ नेचुरल सोर्स एंड द अदर वन इज एंथ्रोपोजेनिक सोर्स. गुड, वेरी, बहुत अच्छा रिस्पॉन्स है तुम लोगों का. यू ऑल आर फोकस्ड. I can see the bright future of our country. टू 2047 में हमारा सपना जरूर पूरा होगा. विकसित भारत. ऐसे स्टूडेंट मिल जाए तो फिर मजा आता है बोलने में, बात करने में. अच्छा है लंच के पहले आपने रख दिया, इसके लिए मैं आपको आभार व्यक्त करता हूं. लंच के बाद तो जमा ही लेते, क्लास क्या लेक्चर सुनते. तो खैर, नेचुरल और एंथ्रोपोजेनिक. एंथ्रोपोजेनिक आप देखो, एग्रीकल्चर में जो फर्टिलाइजर यूज करते हैं, जो पेस्टिसाइड्स यूज करते हैं, इरिगेटेड एरियाज, इंडस्ट्रीज से बहुत सारे केमिकल्स, पेंट्स, टनरी और भी बहुत सारे सोर्सेज हैं. नेचुरल सोर्सेज, वेदरिंग ऑफ रॉक्स, मेन जो सोर्स है वो तो नेचुरल सोर्स ही है, एंड वॉल्केनिक इरप्शंस. ये दोनों सोर्सेज से द आर्सेनिक गोज टू सरफेस वाटर एंड देन इट इज इन ग्राउंड वाटर. और फिर उससे जब हम पानी निकालते हैं, जब उसको पीते हैं, तो हमारे बॉडी में आर्सेनिक जाता है. आर्सेनिक जाना ठीक है, लेकिन बार बार आर्सेनिक का जाना, जिसको बोलते हैं बायोलॉजिकल मैग्निफिकेशन, इट इज मैग्निफाइंग. व्हेनेवर यू आर टेकिंग, एवरी टाइम इट इज गेटिंग स्टोर्ड. बॉडी हैज अ लिमिट. कितना डिटॉक्सिफाई करेगा? So that is the. Now if you see how we are getting exposed to arsenic, these are the environmental sources. The soil, water, air is contaminated, and then the route of human exposure, how it is coming inside our body. हमारे body में कैसे आता है? Through ingestion. If you take contaminated water and food, or inhalation, dust, fumes, dermal contact. If the soil and water is having arsenic, तो dermal contact से, then it is absorbed in the body. Body के अंदर absorb होता है, and it is distributed inside the body. Then it goes to skin, liver, and nervous system. These are the target organs in our body where arsenic goes and accumulates. Then what it does? करता क्या है ये जाकर के? Oxidative stress, DNA damage, enzyme inhibition, and epigenetic alterations. All this is mechanism. This is the mechanism arsenic uses. And then